Thank you very much for inviting me to this virtual event today. My name is Cyrille Delley. I'm a postdoc in the University of California, San Francisco, in the laboratory of Professor Adam Abate. We are dealing mainly with microfluidics, and I'm talking today about my most recent single-cell sequencing method that I developed, and I showcase some applications in cancer biology and in infectious diseases. So if we think about cancers, cancers are genetic diseases driven by somatic mutations, and through the efforts of large-scale sequencing consortia, we now know the genetic underpinnings of about 95% of the common cancers that we encounter. So this puts us in a situation now that we basically know the genomics of the cancers, but we need to find a way to translate this wealth of information into a better understanding of the biology of these cancers and especially into clinical benefits. So what's the challenge in that? Well, cancers are incredibly heterogeneous tissues composed of multiple distinct cell types and different cell clones. And if we now take an average measurement over such a cancer tissue, we essentially convolute the signals from all these cells together, and therefore such a profile is not very representative for the underlying biology of these cells. To solve this is conceptually very simple. We can simply disaggregate the tumor tissue into individual cells and then look at them independently, and that should give us a much better view of how these different clones act and what their biology is. So how can we do that? That's where droplet microfluidics re-enter the scene. Droplet microfluidics is just a tool that's very efficient at creating small compartments. And so we can use these droplets, these compartments, in order to segregate individual cells and therefore isolate them from each other for the analysis. Now, if I talk about droplet microfluidics and single-cell analysis, most of you probably will immediately think of single-cell RNA sequencing, since that has been risen to great prominence over the past couple of years. For this reason, I won't explain it in detail because I think most of you probably know how it works roughly. Just very briefly, conceptually, again, we encapsulate single cells in droplets. We add them together with a so-called barcoding bead. The barcoding bead brings in a primer with a unique droplet-specific barcode, and this allows you then to create a cDNA library using this barcoded primer. If you sequence the collection of all these cDNAs, you can then use this barcode to reassociate the RNA profile with a particular droplet or with a cell of origin, basically. However, I went down this path to single-cell sequencing because we wanted to understand how a cancer genotype drives its physiology, and which essentially means the first thing we need to achieve is to get a measurement of the single-cell genotype of such a cancer. Now, in principle, you can do that with RNA sequencing. However, that's really not ideal because due to a number of technical issues, RNA is just a very poor shadow of the information that is present in the genome. So you really want to look at the DNA of these single cells. But the problem with looking at the DNA in single cells is that the genome is compacted into the nucleus. It's bound to many proteins, chromatin, nucleosomes, and all of this creates an incredibly inhibitory environment for molecular biology, basically the reactions we need to perform in order to barcode these molecules for the single cell readout afterwards. Now, the standard way of dealing with such with the chromatin in DNA extraction is to just use a very harsh lysis condition. This essentially strips the DNA from all the protein components and makes it available for the reaction. However, in this case, the harsh lysis condition is in itself very inhibitory to the molecular biology that we would want to do in order to barcode the DNA. And so the way the Abate lab solved this issue was by using Proteinase K. So what we can do with Proteinase K is we simply encapsulate the single cells together with Proteinase K solution, and then we incubate these droplets of chip for a while, which gives the Proteinase K time to digest the proteins, thereby liberate the DNA, and then we can heat inactivate Proteinase K. And this basically provides us an off switch to turn off these harsh lysis conditions. We then harvest or use these droplets and re-inject them in a second device. So you see on the right the droplets coming in from the top, containing the cellular lysis, and they are paired with a secondary droplet that is just formed on the chip, containing again a barcoding bead and a PCR reaction mixture. We can then harvest these droplets again. These droplets contain the said barcoding beads. So the barcoding bead is a scaffold, bringing in again the primers. Each primer features a droplet-specific barcode sequence, and then a targeting moiety that targets one of up to several hundred cancer hotspot genes that came from these consortia sequencing experiments. So we target all of them simultaneously. So in essence, what we have in these droplets is therefore a multiplexed PCR that simultaneously amplifies the genotype of these cells and provides a single cell barcode. We can then, after the PCR, harvest again these droplets and extract the amplicons. And what we would like to see is equal amplification of each of these targets in all the cells and over all the targets. And so what I'm plotting here is on the x-axis the different targets, about 50 that we were targeting in this case, and the box plot gives the distribution of the amplicons found in the individual cells. And what you can see is that this distribution is very level, indicating equally well amplification of most of these targets. And that ensures that we have very high sensitivity in detecting the genotypes, and it gives us the possibility to resolve the haplotype information, since we will be seeing both copies of the genome simultaneously with high probability. So what does that give us at this point? Well, that's exactly the same thing as what people would be doing if they do bulk sequencing, targeted bulk sequencing of cancer cells. But through the barcodes, we have this now at single cell resolution. And I guess for those of you who have heard of Mission Bio, which is a startup coming out of the Abate lab, they basically recently commercialized this microfluidic workflow as what I have described here. So what can we do with these kinds of measurements or experiments? Consider this patient with acute myeloid leukemia. This is a fish plot with three different measurements. On the left, we have a diagnosis sample. In the middle, where the fish is thin, there is a remission. And on the far right, we have a relapse. So if you look at the diagnosis sample, you can see color-coded, there are 4 different clones present at the original sample, each with a distinct genotype. Now, if you look at the right side at the relapse, we see that the clonal compartment has changed dramatically compared to the diagnosis sample. And in particular, we note that the majority of the cancer cells are now composed from these red clones. And in fact, this red clone is already present in the beginning at the diagnosis sample with a very small proportion of cells. And this basically indicates that this clone was never effectively treated with the treatment regime that was chosen in this case and seeded the relapse. Now, this I find very interesting, but what I really would like to know is, why do these clones await treatment? What makes them special, really, to succeed at seeding this relapse later on? And to understand that, I think the genotype is simply not enough information, because we basically already know from the central dogma of molecular biology that although DNA contains all the information, it's translated into the cellular phenotype in protein space or even in functional space through multiple regulatory events, and all of them influence what will be retained or will arrive on the right end of this information flow. And so if you really want to understand the phenotype of the cells, it's probably not good enough to just measure the genotype. What we need to do is correlate it with a phenotypic readout. And so what we decided to choose as readout is the protein space for two reasons. First of all, it's on the far right of the diagram, so it incorporates most of the regulatory events are upstream, so all of these are incorporated in at this stage in the sort of the information flow. And second, proteins is the interface that is amenable to treatments. Most of the drugs act on the proteins, so that's probably clinically the most relevant aspect of the phenotype in some sense. So how can we measure single cell proteins at the single cell level? A very classical way of doing this is by using a fax machine. So the idea is you label antibodies with fluorophores, you can stay in cells with these antibodies, and then you flow these cells through a fax machine, which allows you to detect the fluorescence profile for each cell, which can be translated in an immunoprofile. And if you want, you can sort these cells. And so this would, in theory, allow you to, for example, sort out a distinct immune compartment, immunophenotypic compartment, and sequence it in order to associate it with a genotype. However, this seems very cumbersome, and it seems it can only really work if there is a tight connection between the immunophenotype and the genotype. Then that's basically the only way such an approach could resolve the connection of phenotype to genotype. So we thought, Adam and I, how can we make that better? How can we have a more general solution? And so what we thought of was DAb-seq, a method that the Abate lab has previously developed. So the idea behind antibody sequencing is that you can simply swap out the fluorophore and antibodies for a DNA barcode. And now, if you use these barcoded antibodies to label single cells or cells, this allows you again to switch out the fluorescence detector of a fax machine for a sequencing machine, and therefore convert essentially the signal into sequencing space. Now, with that idea in place, it's very straightforward to come up with a workflow that can profile single-cell genotypes and immunophenotypes simultaneously, because all we need to do is to stay in the cells with these DNA-tagged antibodies and then apply them to the microfluidic genotyping workflow I just described previously. If we then harvest the droplets from this workflow, what we receive is again a multiplexed PCR in droplets. The aspect of the genotype is exactly the same as I described before, but in addition, we get a second reaction that does barcoding with another primer on the antibody barcodes that are present in these droplets. And so after sequencing, we receive two kinds of matched matrices. Both are the cells are in the columns, and then we have a categorical vector for the genotype calls. The genotype can either be wild type, heterozygous, or alternate for any measured position. And the antibodies are just count vectors where we have the detected number of antibody barcodes for each antibody and each cell. Now, because we can measure many more antibodies than what you can resolve on a fax machine, we can go up to hundreds. It's somewhat cumbersome to interpret this data in a similar way as you would interpret a fax result. And therefore, we use summary statistics or dimensionality reduction algorithms, such as, for example, the uniform manifold approximation and projection algorithm, in order to analyze and investigate the data. And so just to give you an intuition behind this, how UMAP works, the idea is it's basically a probabilistic map that attempts to map cells that are close or similar in their profile in the high-dimensional antibody space into a close neighborhood in the two-dimensional representation on the right. So what we expect to see is that similar cells should sort of visually cluster together in this case. And so what we see here looks like there are 3 such visual clusters present in the UMAP, and that's exactly what we should expect to see, since this is from a proof of principle experiment that I did on cell lines, where I mixed together a myeloid cell, the KF62, a T cell line, and the B cell lines. Since these cell lines have different immunoprofiles, you would expect to see that with the DAb-seq readout, as we do indeed see here. Now, in addition, since cell lines are derived from distinct patients, they inherit the genetic fingerprints of these people, and therefore we should be able to map the genotypic differences between those cells on top of this UMAP representation. And that is indeed the case. So as you can see, for example, for this KIT gene, we have 3 different genotypic signatures, one of a wild-type cell, one of a heterozygous cell, and one of an alternate cell. And they map exactly to the immunophenotypic compartments defined by the antibodies of these 3 cell lines. So this essentially proves that the method works. With that in hand, I approached Professor Catherine Smith and Cyrille Perez. Both of them are hematologists at the UCSF, and asked them if they would be willing to collaborate on a small case study to see if we can learn something exciting from some patient samples. And what they have been working on recently was this case where they had a patient that was treated for over a year until it's a case of acute myeloid leukemia. After 1 year, they finally achieved remission, but then unfortunately, about 4 years later, the person relapsed and the cancer came back. And so I analyzed these last 4 time points indicated here in blue. So at the top, you again have a representation of a fish plot, and what we can immediately see from the DAb-seq results is that the clonal compartment of this patient was composed from a single clone, so only one genotype, with a mutation in the NPM1 gene. Now, if we look at the immunophenotype at the bottom in the UMAP representation, we again get the immunophenotypic compartments for the normal cells on the right, and then an immunophenotypic signature that is characteristic for these kinds of cancer cells. And interestingly, over the whole treatment time curve, so these 4 samples that I processed from this patient, the immunophenotype remained very much constant. So all the time the cells map back to the same compartment. And even more interestingly, we have a very clear separation in the immunoprofile of the CD33 marker between the healthy cells on the far left and the cancerous cells in red. This is particularly interesting, I guess, in this case for clinicians, because there's an antibody drug targeting CD33, and since these cells separate so well, that makes this patient very much amenable to treatment by this kind of drug. And indeed, that's what has been used on this patient on the final time point. It's called gemtuzumab. And as you can see, this puts this person into remission. So that was a great success. Next, I looked at a second patient, which was, in this case, the flow study that clinicians typically do in the course of the diagnosis, uncovered the atypical blast compartment, meaning that the immunophenotype was somewhat uncommon. And so I subjected these same samples to the DAb-seq experimental workflow. And again, you have a fish plot at the top from what we saw on the genotypic side. Again, the cancer clonality was relatively low. We have essentially two clones, one composed out of a DMT3 mutation, and then a subset of these have an additional NPM1 mutation again. Now, if you look at the bottom at the UMAP representation of this initial time point at the diagnosis sample, you see that the cancerous clones in orange are mainly contained in this atypical blast compartment. However, they are also distributed throughout other blast compartments on the top right, and especially also through more healthy immune signatures. And so that's very much different from what we saw in the first patient, because in this case, we have basically no correspondence between the immunophenotype and the genotype of these cancer cells. And as you can imagine, uncovering a situation like that would be extremely hard if you need to do it sequentially, for example, by doing a FACS sorting and then genotyping of these cells, really highlighting the kinds of information we can gain with DAb-seq. Now, in addition, when we followed this patient a bit longer along his treatment history, we see that in the third UMAP, in the relapse sample, or like at the fish plot at the same time point, in the relapse, the clones acquired an additional mutation in the FLT3 gene. FLT3 is a signaling gene mutation, which is relatively commonly acquired in relapsing AML clones again. And as you can see, this dramatically changed the immunophenotypic profile of these cells. And now we have, again, a much more clear-cut relationship between the immunophenotype and the genotype. So this is really interesting for me to see that that there is this discrepancy, especially in the first time point, between the genotype and the phenotype. And it really highlights that tools like these are necessary in order to have any chance to disentangle these complex relationships between the two cell types, between genotype and phenotype. Now, of course, these are just one or two patients, so that's only suggestive and not a statistic. But while I was working on this, I also shared the method and collaborated with a group in Boston who had been working on a patient cohort on 120 or so patients. And in one of the major results, they also saw that typically acquired mutations in the signaling genes, like FLT3, for example, cause a shift in the immunophenotype. So I think that's something that people will probably be following up in the future. Now, as a particular design decision we did in the development of this method was that we basically built it on top of this commercial microfluidic system from Mission Bio in order to enable non-expert groups, non-microfluidic expert groups to use the technology for their own questions. And it's really satisfying to me as an engineer to see that many publications came out over the last year that already picked it up and applied it to their research questions. So for the last few minutes of my presentation here, I switched gears a little bit because it occurred to me that the capability to measure a genotype together with a phenotype could be actually quite powerful outside of the field of cancer biology. I was particularly thinking of infectious diseases and intercellular pathogens, where you basically have a foreign genetic entity hijacking a cell and sort of trying to modulate it for its purposes. And so I was thinking particularly of HIV. HIV is a viral pandemic that has been ongoing for a long time. It still has caused more death than the recent COVID pandemic. And so it seems like a really valuable and interesting target. Now, HIV can be treated with a so-called antiretroviral therapy, or ART. ART prevents the viral life cycle, interrupts the viral life cycle, but it's not a cure. So people need to take these pills for life, basically. And since only about half the world population has access to this kind of treatment, the necessity for a cure is still quite immense. So how does the life cycle of HIV roughly work? So once you get infected, the viral load shoots up, as depicted here in this red curve, and HIV immediately establishes a so-called latent HIV reservoir. So people think that the CD4 T cells are the major cellular component of this reservoir, which is basically integrated viral DNA in the genome of these cells. HIV in this form is very persistent. The antiretroviral therapy can target the red part, the viral load, but not the reservoir. So that stays consistent. And if now people interrupt their ART treatment, the viral load shoots up again and rebounds out of this HIV reservoir. So if you want to treat HIV, what we need to do is finding a way to target the reservoir or to silence this reservoir. What's the problem there? Why can't we just do that? Well, these CD4 plus T cells harboring the HIV virus are incredibly rare. So we think of them as 1 in 10,000 or so is probably infected. Then the HIV in this form doesn't express any proteins or RNA. So there's basically no way how we can sort these cells or identify these cells and subject them to further studies. The only unambiguous marker that these cells have is the integrated provirus, which is again like a DNA copy of the virus in the human genome. This raises the question: can we make an assay that exploits this marker to identify these cells, and maybe in addition learn something useful about the physiology of these cells, or find ways that we could use to target these cells? And of course, measuring such a genotype along with physiological aspects of cells is exactly what DAb-seq does. So for this reason, I designed a panel of targeting primers that are spanning the HIV genome with 10 targets. This is not that easy, not as easy as the cancer targeting panels, because HIV is so variable in its genotype, that in its genome, that it's hard to find a spot where you can place a primer. Nevertheless, there are multiple conserved regions that are useful for this kind of purpose. And so the idea here is that since we have 10 amplicons simultaneously, that increases the probability that at least some of these will work, and it also provides the possibility to distinguish truncated virus, defective virus from full-length viral integrates. I again made a mock system to see if this system could work in detecting HIV cells, and for this purpose, I created PBMC. I used a blood sample, PBMCs, and spiked in 1% of J-lot cells. J-lot is an HIV model cell line with integrated virus. And so again, the DAb-seq readout here shows the immunophenotypic compartments. We get the cell types that we expect: myeloid, CD8 killing T killer cells, B cells, and so on. We get here an additional cluster of cells that indeed associates with a very high number of detected HIV amplicons. The median is 8 detected amplicons from HIV in these cells. And so that's likely the J-lot cell line spiking. And again, since these J-lot cells are derived from different patients than the blood cells, they should feature a different genetic fingerprint, which we can again detect with the human panel that is also present in these measurements. And that's what you see on the right. Indeed, the cell line that is high in HIV amplicons, the immunophenotype high in HIV amplicons, is also positive for these genes, like different at these genetic markers. Again, reinforcing the notion that this is the J-lot cell line and the method works. I then reached out to Professor Iain Clark at the UC Berkeley and Professor Eli Boris to participate in a study on patient samples. And so what we assembled are 7 patients from the Raven cohort The ribbon cohort is a cohort of HIV patients on long-term ART. That means they had effective viral suppression for over 2 years. And so we aim to profile some 100,000 cells per patient in order to have a decent number of latently infected HIV cells present. Here I give you a sneak peek of the kind of data that we are seeing from these studies. This is the blood compartment from one of these patients. Again, you have the expected blood compartments. We see more CD4 T cells in this case than what you saw before in the PBMC sample, since we pre-enriched these cells because we suspect that the HIV is mostly present there. So we wanted to make sure that we give the best shot at identifying these cells. So you can ask yourself, where do you think HIV is in this? If you look at it, if you look at the genomic readout, we detect HIV exclusively in these CD4 T central memory compartment. This is very exciting. It is also what we would expect from what people have been doing previously. But with DAb-seq, we are able to show that in addition to just being here in this compartment enriched, it's basically exclusively there. And we also see that the HIV-infected cells are actually enriched in a special subset of the, which I label here, the central memory compartment type 2, which is even more enriched in HIV cells. So I think this is really exciting, and it will be very exciting to follow up on this and see if we can detect any relevant markers for these kinds of cell lines. So with that, I basically conclude my discussion for today. I hope I convey to you that this is a really exciting field to work in, and that there's plenty of opportunities for method development and microfluidics in order to make a dent in the analysis of these challenging diseases. It's very exciting to work in this field where so many different expertises meet. I want to thank especially my mentors, Adam Abate and Iain Clark, and also my colleagues in the Abate lab, especially Ben and Harish, who helped me in a lot of these experiments. And I already acknowledged my fantastic collaborators throughout the presentation, and on the right there are some support and funding sources. Thank you very much.